So we're trypsinizing. When you open these pipettes, make sure you always break away so that you don't touch it, and then try to avoid touching this as much as possible. So what we're going to do is we're going to wash twice with PBS and then trypsinize these cells. So first we're going to remove the media. And now we'll just spray PBS on there. 10 mils of PBS down inside without touching the pipette to anything, so I can put it back in here. Now when I do have to touch it, I just touch at the very top to avoid any contamination. So I'm going to swirl this around and then remove it. And do it again. Now notice when I spray things into the onto the plate, I tilt it sideways and spray down so that it doesn't spray along the bottom and remove any cells that are already there. Now I'm just going to swirl this around again and remove the media, but first I'm going to prepare my trypsin. So I'm just going to remove that. And so I'll just remove that PBS. And add 2 mils of trypsin. Now this one, because it's going to remove the cells, I'm going to spray it directly onto the bottom. There's also not too much volume, so I can't shear the cells off. So now I'll just switch this around, make sure it covers the whole plate, and we'll put that in the incubator for 5 minutes. So now after the 5 minutes is done, we're going to take that out. So if the trypsin worked, you'll be able to see a little bit of foggy cells in there as you swirl it. So now what we're going to do is remove this with 10 milliliters of PBS. So I'm going to do several washes where I spray everything down to the bottom, suck it up, spray it down. And this is also going to serve to resuspend all those cells so that we can count them. So I take 10 milliliters, and I'm just going to stick it inside because I'm not going to use this pipette again. So I spray it along the bottom there, suck it up again, spray it along the bottom again. And this is just to remove any cells that are caught in corners or that might not be all the way removed from the plate. So I'll do this a couple times, 4 or 5 times, just to make sure I get as many cells as possible. Now, if you can see kind of a cloud inside the media with the cells, you should pipette it up and down like this a couple times, just to make sure that all those cells are resuspended. And then we'll dispose of this pipette because we are done with it. Just put that in the biohazard. Now this is also garbage. However, we'll need to know our cell 9, our initials, and then this is passage 10. But because I've just trypsinized it, we'll now label a plate that's passage 11. So now that's labeled for passage 11, which will be the next set of cells that we'll do. And we'll set that aside. Now, so if you notice here, we've got some droplets on there. So to make sure that the concentration is all the way even, we'll just invert this a couple times, make sure the cells are resuspended, and also that we have all the media we can save there. Then we'll take 10 microliters out, and count that. So those 10 microliters will count on the cytometer, and this we'll put in the centrifuge. So we'll put those in opposite of each other. We're going to centrifuge it at about over 3,000 RPM for 5 minutes. So we'll just go ahead and close that and start it. So we're going to take those 10 microliters and we're just going to inject it in here, right, without lifting up the glass. And capillary action is going to cause all that water to cover the surface. So there are 4 quadrants on the hemocytometer, and we're going to count all of those 4 quadrants, and then we're going to average what we're going to do. So our total is 121 cells, so we'll divide that by 4, and that will give us our average. Then we times that by 10,000, and that tells us how many cells per milliliter we have. So our 121 total, we'll divide that by 4, and times that by 10,000, which means 302,500 cells per milliliter. Now in the centrifuge we have 12 milliliters, so we'll times that by 12, which gives us a total of 3,630,000 cells. Now we want to leave that down to 1 million cells per milliliter, and then we need a final dilution of about 100,000 cells per milliliter in order to plate out. So we'll divide that by 1 million, which gives us 3.63. Now I usually ignore this last number, so I will just dilute it into 3.6 milliliters of media. If this 5 were greater, then I would round up. So before we dilute this in 3.6 mils of media, I'm going to use this before I touch it to cells so that I don't have to get out a new one. So firstly, I'm going to plate out 1 plate. So you usually need about 3.5 milliliters of cells at 100,000 cells per milliliter to plate out on this plate. However, if you're just doing 1, you should use about 5 milliliters just in case. So because we have 1 million cells here, and this is a 1 to 10 dilution to get to 100,000 cells, we're going to use 4.5 milliliters of media, and we'll just pipette that into here. Now because this is sterile, I can touch it. However, once I put cells in there, I don't want to touch this with anything and put it back in here. And now I'm going to put about 10 mils of media in here. I'm actually going to do 9.8 because I'm going to put 200,000 cells in here, which would give me about a week before I have to trypsinize again. So I'll just spray that in here. And cap that and make sure it's coating the whole bottom. Now this will be what we're going to put the cells in to grow again for our next passage. So we'll put that to the side here. Now, after taking it out of the centrifuge, you'll notice there's a pellet of cells down there. So what we're going to do is remove all this media without disturbing the pellet. So we're just going to remove that media, slowly move down. Right? And we want to, there might be a little bit of water left in there just so you don't disturb the pellet. And now we'll take 3.6 milliliters. So that's 3. So this is our 3.6 milliliters of media. And we're going to resuspend that pellet. So we'll just pipette this up and down a couple times. To make sure that's nice and suspended. Okay. Now I'll make sure that this is nice and empty, and I can store that in here for now with that other media. Now we're going to use the 200 milliliter pipette and the 1 milliliter pipette, or 200 microliter pipette. So we're going to use half a mil for our dilution to put onto this plate. And then in this one we're going to use 200,000 cells to put onto this plate. So we'll just go ahead and take 200,000 and inject that into here. Now we'll just swirl around, make sure the cells are evenly divided, and then that can go in the incubator. Now we're going to take 500 milliliters of this and put that into here, where this pipette was. So that will give us 5 mils of media at 100,000 cells per milliliter. And we can dilute that. Now, we're just going to resuspend that and then put it into this, so we can then pipette it using the multi-channel pipetter into this. Now this will help our cell count be a little bit more even. So let's go ahead and prepare that. So we need 100 microliters in each well. So we want all 8 pipettes there. So now we'll go ahead and resuspend this and make sure that it's nice and diluted. So we'll pipette it up and down a couple times again, just like we did last time. Probably between 6 and 10 times. Make sure it's all mixed together. And then we'll suck all of it up and spray it into our pipetter boat here. So now we'll take our 100,000 and we're just going to pipette that into this. Now I have PBS along all the outside of the rows, and that prevents these cells from drying out. So I'm going to pipette it here onto the second row and second column, and we're just going to do the first 3 rows here. Now this lets us treat with many other things. Now I'm also going to remove all the pipettes except for 3 and pipette those last wells here. Now that's done, and we can go ahead and put that into the incubator. Now with the rest of this, this can just be sucked up into the pump and thrown away. Same with your cells here. This should be thrown away. And any residue that's in here. Now these can all be thrown into the biohazard with those tips that we used as well.